Hey everyone, welcome to week 7. We are going to be going through gram-positive bacilli this week. So we're actually going into an entirely different group of bacteria. So we're all done with the gram-pos cocci. You'll still continue to test on them in the lab and play with them, but we are done lecturing through them. So now we're moving on to all the gram-pos bacilli. So all of these are going to be packed into just this 1 week. So just this week is gram-pos bacilli, and then next week we'll switch gears into something else. So I'm going to pull up the PowerPoint here that you have in your lesson tab. Let me see here. Okay. All right, so we're going to just jump in. First gram-pos bacilli that we're going to start with is the genre Bacillus. So these are aerobic gram-positive rods that form spores. So these are going to be the only gram-pos rods in this lecture PowerPoint that do form spores. All of the rest in this PowerPoint do not form spores, so that will keep them apart from the others. These are also very much catalase positive. The biggest one is going to be the Bacillus anthracis. That is our main pathogen. The rest of Bacillus tend to be very low virulence, meaning they really just don't cause a ton of diseases. We will talk about one other bacillus that can cause a couple things. The rest really don't. So the good news for you is you really only have 2 bacillus bacteria to remember. Bacillus anthracis, which can be found in the soil. You can acquire it through inhaling those spores that it produces. And then the other bacillus, which is going to be called Bacillus cereus, which we'll see on another slide here. Those are going to be the only 2 that really we go through. So that is what bacillus looks like on a gram stain. As you can see, it's very much that rod or bacillus shape. They are very large gram-pos rods, so hopefully that's obvious to you. You hear how much they stand out. A lot of times we nickname them box cars. So when you think of a train, you know, they have box cars, right? And those are very large, rectangular, and that's kind of how they got their nickname here because they're such large rectangle-shaped rods that they can sometimes be nicknamed box cars. On this, you'll notice a little bit of a lighter area where it's not as stained as darkly with the crystal violet. That is the spore. That's where the spore is. So where it has the spores that it produces, they don't pick up the gram stain as well. Now, just a note that you might not always get this beautifully crystal clear picture like this in real life. We all know in real life not everything looks like a textbook. So not always you will see the spore area, but I thought this was a really great representation to show you that sometimes you do get to see it, and this is a good example. Okay, so let's jump into the Bacillus anthracis. Hopefully on that second word, you've kind of already thought to yourself what that sounds like or what that looks like when you read it, and it should look like anthrax. So Bacillus anthracis is what produces anthrax. And now I think we've all heard of anthrax, especially because of bioterrorism and the risk involved there. And it very much is a bioterrorism weapon that is used. But there is another name for anthrax called wool sorter's disease. So wool sorter's disease picked up its name, and I think I have an announcement coming out on this later this week. But basically, because you can find Bacillus anthracis in the soil, you know, and sheep graze grass, right? And when they would go shear the wool off the sheep, those sheep handlers, whatever you want to call them, or wool sorters, would contract the anthrax through the handling of their wool because the anthrax or Bacillus anthracis was in their wool from grazing along the soil. So that's why it's also called wool sorter's disease. There's an entire island, I think it was off the coast of UK or Scotland, that kind of talked about this, and that's the article that I have linked in an announcement later this week. I always think it's just an interesting story, kind of helps keep it in perspective, what it is, where it's found, you know, just to me, it's easier to remember. So what you guys probably don't know is that anthrax actually comes with 3 different phases. So we all think of the bioterrorism one, but there's actually regular infections that can occur in the skin or in the GI tract. So there's going to be 3 phases. So you can either have the cutaneous, the GI tract, or the respiratory. So we'll just go through each one of these 3 phases for Bacillus anthracis. So the first phase is mentioned here, cutaneous. That's where you're getting the Bacillus anthracis spores entering through an abrasion on the skin. That's most likely what the wool sorters people were getting, were the cutaneous form. So that skin infection will be kind of start as an itchy bump that will kind of resemble an insect bite, and then will, in a couple days, develop into a larger vesicle and then into a skin ulcer. So much larger, 1 to 3 centimeters in diameter. Typically in the center, it'll show like a black kind of dying area is how they describe it. Inhalation is the respiratory version of Bacillus anthracis. This is almost always related to bioterrorism. Anytime we think respiratory and it grows out anthrax, we're right away thinking bioterrorism. That's the route that is usually used for bioterrorism because it's so easy to breathe in those spores. So the symptoms will start like a cold. After several days, they'll progress to very severe breathing problems. Your body will go into kind of shock. It's very fatal, which is part of the reason why they love to use it. Not only is it easy to pass to the person by breathing in spores, it's extremely deadly to those people as well, and very much, if not deadly, it definitely takes you out and down, you know, with all the breathing problems and therapy that needs to go with it. So again, like, I don't know, I would say at least 90% of the time, if not more, and I've read some places where it says almost always, if we see anthrax due to a respiratory condition, we right away have to think bioterrorism. There's no other reason for it. To me, though, I don't like to say 100% of the time, but I'm saying greater than 90% of the time, if it is seen with a respiratory kind of deal, you should be thinking bioterrorism. And then finally, the third condition that can be seen is the GI, gastrointestinal, and this is where you're consuming contaminated meat, and then it gives you kind of like a food poisoning case. So those are the 3 different phases of infection for anthrax, or what we can call wool sorters disease. All right, the other bacillus that I discussed that we need to know is Bacillus cereus. The 2 diseases I want you to link here is food poisoning and eye infections. A lot of times with the food poisoning, it could be just the spores that were left over because, again, Bacillus does produce spores. Or in the case of Bacillus cereus, it very much produces a neurotoxin, and that neurotoxin can be left behind in contaminated meat, and if you consume that, that will still give you the food poisoning. You don't always have to have the bacteria present to get the disease. It can leave behind its toxin, and that will still do just as much damage to you. All right, so to grow bacillus in general, to grow Bacillus anthracis and Bacillus cereus, of course, sheep blood and chocolate work great. They grow pretty much everything, right? Thioglycolate broth is also another really good supportive broth, so that will also grow it. Now, these are gram-positive rods, so they will not grow on MacConkey agar because MacConkey grows gram-negatives. Bacillus can grow a little bit on PEA or CNA agar because those do grow gram-positives. Now, they don't always grow. Sometimes they grow, sometimes they don't. It's not like crystal clear all the time, but just kind of a mention. So the best use here would be your sheep blood chocolate agar since those are supportive. So what do they look like? Very important to take away their colony morphology, especially with anthrax. As microbiologists, we have to always be aware of the list of nasty organisms that are like our naughty list of organisms that we're always screening for, because the minute we think we have anthrax, that better be sent out right away to your State Department of Health to be worked up, to be, you know, that kind of thing. We're taking this very seriously. So we always have to have in our mind what these look like, any suspected kind of cases. But what bacillus looks like in general, they're very large colonies. Both cereus and anthracis are large colonies. Bacillus cereus in general is called a spreading colony, just very big colonies, probably gray to white in color. Bacillus anthracis is also large. This is the picture here is Bacillus anthracis, just so you know. And they are said to have medusa head or comma-like projections. So for those of you who don't know, Medusa is the lady in Greek mythology that had snakes for hair. And I don't know who came up with the term Medusa head. I think the term comma projections, like a comma, is more adequate. What essentially that's referring to is that these are not perfectly round colonies. They always have some sort of projection coming out of them. They're not going to be perfectly round. So when you look at the picture here, you can kind of start to see how sometimes they have those little projections on a few of those. I know they're not really well isolated to see them, but that's what they're referring to with the anthrax. The other big thing with Bacillus anthracis, and you can see this with the one colony that I'm trying to circle with my mouse, hopefully you can see that, is that when you go to take your loop to it, they're very sticky. So they'll kind of stick up or string up with the loop when you go to pick it up, and that's kind of what it shows here, is that the loop kind of touched it and then it would stand up and kind of stick up. So very sticky. All right, so to identify, so on the auger, we didn't mention this, but part of the colony morphology, Bacillus anthracis is non-hemolytic, but Bacillus cereus is beta hemolytic. Bacillus anthracis is non-modal, Bacillus cereus is modal. They are both catalase positive because all bacillus are catalase positive. Bacillus anthracis is penicillin susceptible, so if you were to run an antibiotic susceptibility test, it should be penicillin susceptible. Bacillus cereus is penicillin resistant. They are both positive for an enzyme called lecithinase. This is basically looking for a zone around the colonies on an auger called egg yolk auger. So it's just a specialized test that you would have to have in your lab to run. Again, not every lab is going to have this. So the nice thing is you can see how these are definitely differentiated. And I always just try to remember the nons go together, non-hemolytic, non-modal with anthrax. And then, and this might sound silly, but in my head, I always remember it's penicillin susceptible because it amazes me that you have this big bad disease called anthrax that scares the bejesus out of everyone, but you can treat it with penicillin, which is like the first antibiotic ever made. It's like crazy to think that this big bad disease can still be treated, hopefully, you know, caught in time and treated with penicillin. So that's kind of what I link in my head. Use whatever works for you. Just know the test results for each one so you can separate them apart. Other lab identification, Bacillus anthracis does have a capsule, so you can look for that capsule with India ink stain. India ink stain, you will hear this mentioned again with a yeast called Cryptococcus when we get into the mycology chapter, which is in micro 2. But basically, it's like this kind of a brown stain, and so when you stain it, everything stains brown except where the capsule is because the stain cannot get inside the capsule. And so it kind of creates like a halo effect. That's what it does with the Cryptococcus as well. So that's kind of how India ink stain is used, is it will just kind of stain everything brown except the capsule. So they can use that part of it. You can use a special media that has sodium bicarbonate in it to look for the capsule as well. So sodium bicarbonate will help also detect the capsule with that media. And then again, an increased carbon dioxide setting as well will stimulate the capsule production. So any of these three will help detect that to help identify Bacillus anthracis. If you want, and there's another test where you induce sporulation, induce spore formation basically, because these form spores, and you can do that with urea agar. So I would definitely know these as well. Treatment, again, Bacillus anthracis is still very much susceptible to the beta-lactam groups of antibiotics to include penicillin. Bacillus cereus is penicillin resistant because it forms that beta-lactamase enzyme that hydrolyzes the beta-lactam rings, so those antibiotics do not work on it. Okay, so that's it on Bacillus. We're going to move over to our next couple groups of Grandpa's rods, which are Cranibacteria and Listeria. So again, both these groups are also catalase positive. They do not form spores. Like I said in the beginning, only the Bacillus group forms spores. So they are also catalase positive though. These Cranibacterium Listeria are pretty much found in the environment. They can be found as normal flora on people, obviously humans, and they can be sometimes associated with animals. The two main species that we're concerned with are Listeria Monocytogenes and Cranibacterium Diphtheria. Hopefully you should recognize that second word there, diphtheria, for diphtheria. There is some others that are opportunistic. One good example would be Cranibacterium Diphtheria. Sometimes that pops up here and there as a problem, but for the most part, we're really concerned with these first two that we listed. All right, so let's discuss Listeria Monocytogenes. This has the capability to survive within phagocytes, so it helps keep it surviving. Very much found in nature through soil, vegetables, animals, that kind of thing. Sometimes can colonize in humans, like in the GI tract. The big problem with Listeria monocytogenes is that it can be passed from the mom to a fetus that she is carrying, especially if you were to ingest Listeria, it very much is problematic to that fetus especially. So the thing that it mainly causes is meningitis, and that's why it's super important to protect that fetus, because it will do neonatal meningitis. So that's the big disease listed with Listeria Monocytogenes. Just a note on its virulence factor, it does have some toxins it can produce that will help allow it to survive those phagocytes. Grow it again, blood agar and chocolate, because those are both supportive, and they will be smaller beta hemolytic colonies on those two agars. The big characteristic with Listeria monocytogenes is its motility. It is extremely motile, very characteristic patterns that are not seen with anything else. So if you were to do like what we call wet mot, where you basically just take a drop of like saline or liquid that has Listeria mixed into it, take a drop of that, put it on a slide, put it under the microscope, you should be able to see what they call a tumbling motility that happens. So basically kind of like a somersaulting or end over end to the rods, they will be moving like that, and that is so characteristic. Nothing else has that but Listeria monocytogenes. There is a different motility test that you can run that's run more in an auger kind of tube, and that will take an umbrella shape. So it'll actually have the handle of the umbrella with the actual umbrella top to it. So those two are so characteristic. Such big items to remember for Listeria that it sets it apart from anything else. A couple other tests, it is CAMP positive, it is hipurate positive. So at this point, you should be thinking, gosh, some of these tests sound really similar to Streptococcus agalactiae, group B strep. Remember, we talked about group B strep, how that was CAMP positive, hipurate positive, and that also was beta hemolytic. It also caused neonatal meningitis. So they're similar in that way. They both are beta hemolytic, both CAMP, hipurate positive, and both cause meningitis in fetus. How you separate them, though, is, remember, is strep, besides the gram stain, is that strep agalactiae group B strep is catalase negative, and Listeria is catalase positive. And then again, Listeria has these unique motility reactions. All right, so that's just showing it was supposed to show, the right side is showing a CAMP test. The left side, I think, was supposed to show the beta hemolysis, but that's a bad picture, so just ignore that. There is a YouTube video. I'm not sure if this still works or not, but you can check it out if you'd like, showing the umbrella motility. It literally will take, this is like the handle, and then eventually it will get the umbrella shape at the top of it, and that will show that umbrella motility that we discussed. The only other thing before I move off of this to be conscious of, like I said here, you know, moms have to be really careful from getting this from ingestion. A lot of times this is found with like homemade cheeses or softer cheeses, certain, I think, deli meats. So there's certain foods that are really kind of told not to touch at all when you're pregnant. So you can look up listeriosis on like the CDC website, and it should talk about that, those kind of foods that they should be aware of and kind of stay away from, so that they prevent picking this up. There's been several times where this will hit the news that somebody has had this or whatever it might be. All right, Cranibacterium Diphtheria is our other big pathogen out of this chapter. This is carried by humans. It is never considered part of their normal flora. It only is maybe carried, but not really part of them. It was transmitted very much through respiratory droplets, or of course, direct contact with anything that might have been contaminated. Obviously, it causes diphtheria. That's why it's named Cranibacterium Diphtheriae. There is two forms of diphtheria. There's a respiratory form, which is the more deadly form. That's why we all have a vaccination against it. Because it blocks the airway, and that's how it becomes really deadly. But there's another form that's just a cutaneous kind of skin ulcer forming diphtheria. So Cranibacterium Diphtheria in general has an exotoxin that will help destroy host cells so that it can survive. So a little bit on the respiratory diphtheria. Again, it starts kind of like a pharyngitis, which is a sore throat. It develops this pseudomembrane on the back of the throat, and that membrane will cover over the tonsils, the palate, the pharyngeal wall, and basically results in covering over the airway and resulting in that respiratory obstruction. The toxins of diphtheria get released into the bloodstream and can also cause damage to the other organs. And in general, all of this adds up to become very life-threatening, which is why they were able to thankfully get a vaccination developed so that we can all not have to worry about diphtheria. So this is showing the membrane in the back of the throat. Do you see how small that airway just got there, how obstructive that is on that in the back? I mean, there is like no room there to breathe. All right, and then again, it forms big, nasty skin ulcers in the form of cutaneous diphtheria. And we don't need to say more on that. All right. So again, these are gram-positive rods. These are very neat, though, on a gram stain. They take some unique arrangements on a gram stain that no other gram-pos rods have. They are said to be referred to as Chinese letters. So you kind of know how Chinese writing looks, and they're said to have all those kind of patterns like Chinese letters, or they're seen in the shapes of the letter V or L, or they will be palisading. What that means is they're lined up parallel to each other as rods. So they're lined up, each one parallel to each other. So very unique arrangements under the gram stain. So definitely remember that. That's very characteristic for Cranibacterium diphtheriae. So this is showing you the Chinese letters. I think that's just so amazing how they look. Yeah. Okay. There is some specialized media that is used to grow Cranibacterium diphtheriae. If you ever suspect somebody were to have it, this will for sure grow that diphtheriae. So we have cysteine telluride agar, we have Leffler agar, which maybe isn't used as much anymore, but you should still be aware of it. And then Tinsdale agar is a great one. This one has, do I have it on the next slide? Yes, it will grow as a black colony with a brown halo. Very, very characteristic for Cranibacterium diphtheriae. That is a nice test to help identify it. So Tinsdale agar will have that black colony with a brown halo. So on blood agar, these tend to be just smaller, gray. Sometimes they can be beta hemolytic, sometimes not. So they really don't have anything unique on a blood agar plate. It's just when you start like gram staining, testing, you kind of start, and then you're going by the patient's symptoms that they're showing, you kind of start going, oh, you know, it could be diphtheria, you know, you kind of put it together that way. And that might be urging you to say, oh, let's sub this out in Tinsdale agar, let's sub this out in cysteine telluride agar, you know, that kind of thing. So this is what it looks like on that Tinsdale agar with the black colony and a brown halo. All right, so a little bit of a note on Cranibacteria in general. You will hear a term used in the microbiology lab called diphtheroids, or sometimes you can call them coriniforms, which is bolded there. But diphtheroids is a term that's used a lot to describe other Cranibacteria. There's other species of Cranibacteria that are low virulence, don't really do much, and they're very much normal flora on our body. So there is other Cranibacteria that are normal flora of like the skin, and they're not the true pathogen diphtheriae though, but they're just called diphtheroids. So if you were to do a blood culture and you picked up a little bit of that skin flora and say in one of the blood culture bottles you found a Cranibacteria gram-pos rod, I would not call it out as diphtheriae, because if it's in only one of the bottles and not all of them, it's probably just contamination from the skin. Or sometimes you'll see it show up as contamination on an agar plate. You'll gram stain it, you'll see it's a gram-pos rod, but it doesn't really go with the source you're looking at or whatever it might be. So just take a note, we're really concerned about Cranibacterium diphtheriae if we're seeing it isolated from a very sterile site, something that should never have bacteria, if we're seeing it show up in multiple blood culture bottles. Or if it's a truly pure culture, no other organisms on there, because then we know, yep, this is definitely a pathogen, not just contamination. So again, there's lots of Cranibacteria diphtheroids that are normal flora. The only time we're concerned with Cranibacterium diphtheriae if it meets one of those three items that we just discussed. So that's kind of just a tidbit on that. So other things that are going to help differentiate Cranibacterium diphtheriae from the other Cranibacterium besides the Tinsdale agar, it is very much urease negative, whereas the others tend to be urease positive, so that will also help. And to really identify it, usually if you get Cranibacterium Diphtheria in your lab, you have to send it to your State Department of Health. It's one of the ones that they want sent to them because it's something they track since it's so heavily vaccinated. Anybody that's not getting vaccinated, then maybe it shows up, but we really need to be careful. This is something we're tracking the presence of. You know, whenever you hear of outbreaks of, like, you heard of the whole outbreak of mumps at Disneyland, well, that's because they're tracking that through the State Department of Health, and it reports to the CDC. Same thing with diphtheria. They're always tracking whenever it shows up positive. So we would send it to a reference lab and ultimately to the State Department of Health, and they're going to look for the diphtheria toxin there to really say, yes, this is truly proof. Of course, to prevent it, hopefully we're all vaccinated through our Dtap vaccination. The D in Dtap stands for diphtheria. If you've been exposed, there is different antibiotic treatments that are available. And then just a note on Cranibacterium Diphtheria, because that is an opportunistic pathogen. Again, it can be found as normal flora in the skin, the rectal site. It's not very virulent, but if that person is really immunosuppressed, hospitalized, it will take advantage if it can. It will cause infection if it's allowed to. Different wound infections might pop up at like the IV device site, or septicemia if they're really down and out and have no immune system to fight it off. That's when it will take advantage. So this is just kind of a note on the gram stain, on the appearance, on the colony morphology. We've already discussed all this, but it's just kind of nice and summarized right here for you. And then a flow chart. We flow chart almost everything in the lab on how we work up. We've already discussed Gram-Pos Coxy flow charts lots. So this is just kind of a note on how you can perform a flow chart for Gram-Pos rods. Now again, every lab is different on what testing they choose to use, but if they say, this one's specific to Cranibacteria, if they get a brown halone tinsel agar and it was a Gram-Pos rod and it was urease negative, then it's probably Cranibacterium Diphtheria. So they put the two together. If it was urease positive, then it's probably something else. So they just show how they can work up Cranibacterium. Okay, some other Gram-Pos rods. We're going to start with our, well, they're these three. Gosh, I cannot talk. Erysipelothryx, Garderella, and Lactobacillus. They are all catalase negative, non-spore forming Gram-Pos rods. So all of the rest of the bacteria we've talked about so far have been catalase positive, so these are all catalase negative. These are not very big, large pathogens, but they definitely are something that can come up, so we want to just discuss the one disease linked with each one. Erysipelothryx rhusiopathiae is often part of animals. It's carried by animals, so anybody that handles animals a lot could pick this up, especially if they have like a wound or a skin abrasion that it can get into and infect that wound. So Erysipelothryx rhusiopathiae very much causes skin infections through handling of wounds or being exposed to animals, whatever, handling wounds, handling of animals or being exposed to animals and getting it through something on their skin. I know when I learned in college about this bacteria, it always stuck in my head about pigs. Pigs are really big, can be really big carriers of this organism, but so can other animals. Garderella Vaginalis is, just like the name says, it's normal vaginal flora, and it will cause bacterial vaginosis, so a bacterial vaginal infection. And then Lactobacillus actually doesn't cause infections. It's extremely rare. It's actually a really good bacteria. This is a bacteria we have in our yogurts that a lot of times, you know, that's why they want to eat yogurt, to get some of that good, healthy, normal flora bacteria back, especially if somebody has had, like, say, a yeast infection or some sort of vaginal infection and they wiped their normal flora clean down there through antibiotic use or whatever. Let's get some of that back in there. We need to rebuild that normal flora. There's a reason for it down there to help prevent other infections, things like that. So, again, they're all low virulence, but Erysipelothryx, again, related with skin infections, and they're specifically called erysipeloids, the type of skin infection that Erysipelothryx gives. Garderella vaginalis, like I said, bacterial vaginosis. Sometimes you might see it show up as a UTI. And then Lactobacillus, typically contamination, extremely rare for it to cause anything. You'd have to be extremely immunosuppressed for this to come up as anything. They will all grow on, of course, sheep blood agar, chocolate agar, because those are supportive. Again, sometimes grandpa's rods will show up on CNA agar. There is a special agar called tween agar that will help grow Garderella Vaginalis if we ever suspect that. And one note on Erysipelothix rhizopathia. One of the big characteristics of this is it will produce hydrogen sulfide, which is called H2S. That is a black precipitate that it forms. So if you were to put this in an agar called triple sugar iron agar, this is an agar you'll hear more of next in micro 2. We use this more with gram-negative stuff, but it will form this H2S black precipitate, which you can see at the bottom of the tube here. That black is the H2S. This is the only grandpa's rod that produces H2S. So it's a very unique characteristic for Erysipelothix. As far as Garderella goes, I'm not sure if you guys have taken your analysis or not. If you have, and I can't remember if where they cover it in there, but you might have heard of something called clue cells. Clue cells are basically those squamous epithelial cells, which are just off the skin, and they are filled with the Garderella Vaginalis bacteria. And these are very characteristic for bacterial vaginosis infection. So sometimes when a woman comes in and has signs of it, the doctor will do what we call a wet prep or wet mount, whatever you want to call it. But basically they will swab the vagina, send the swabs to the lab. We will put it on microscope slides and look at it under the microscope. In there, you will see clue cells. You can also see clue cells sometimes in urine samples because remember, it can sometimes go with UTIs as well. So when you see these clue cells, you'll see that they're large squamous epithelial cells, but they don't look quite right. They look a lot more filled up with something. They're really refractile. So if you like focus up and down, they're kind of shiny. And then the borders of the cell will be really obscure. Those bacteria will kind of cover everything, so you're not going to see a nice delineation of the border like you normally do. So this is kind of what it looks like. You can tell that these are large squamous epithelial cells, so nothing big, those are skin, but you can see that they're definitely filled up with something. They're not clear here like they normally are. And then these borders are very rough and not very nicely outlined like they typically are. And again, if you're to focus up and down, there should be a little bit refractile. Okay, branching grandpa's rods. So this is considered Actinomyces. These are all grandpa's rods. They are what we call branchings. So they'll kind of form filaments on the agar. So they'll either be along the agar surface or they'll extend up into the air or even dig into the agar. So they kind of have branches up in the air or along the surface. They can be, usually they're aerobic or facultative anaerobes, meaning they don't care if they're anaerobic or aerobic, so they can grow either way. They're not frequently seen, so this isn't something you're going to hear of much. I've never seen one myself in person. Hopefully your lab instructors have, because they would be more experts at describing what they have seen. But when they are seen, they can cause some very serious disease, but they are hard to isolate. So typically, acid fast isn't something we've talked about a lot. We've discussed this earlier when we talked first about gram staining. Remember, acid fast was that stain that targets mycolic acids in the cell wall, and this is for bacteria like mycobacteria. We talked about that mycobacteria have mycolic acids in the cell wall, and so we would acid fast stain them and not gram stain them. There is a few of these organisms in this group that also will be acid fast because they because they have mycolic acids in their cell wall. And then there's another group of actinomyces that are non-acid fast, that are just normal. You can gram stain them. And when you do gram stain them, you will see their grandpa's rods. So here is the different ones that fall under each group. So in the acid fast, you're going to see Nocardia, Rhodococcus, Gordania, Sucamorella, and then ignore the Cranibacterium. We don't care. We've already discussed that. Just ignore that. I should delete that. The one that I really want you to know out of the acid fast group is Nocardia. I will not test you on any of the others or talk about the others. So Nocardia is the main one I want you to take away out of the acid fast group for the Actinomyces. Out of the non-acid fast group, the only one I want you to know there is Streptomyces. So for the case of Actinomyces groups, you need to know Nocardia and Streptomyces. So far what you should know about these are their gram-pos rods that are branching. Nocardia is acid-fast because it has mycolic acids in the cell wall. Streptomyces is just normal gram-pos rods, non-acid-fast. So we're going to start with the Nocardia. It is catalase positive. This one is a strict aerobe. It does need oxygen to grow. So there it's showing the positive acid-fast reaction of Nocardia. Again, when acid-fast is positive, it'll be that bright pink color. Here it is on the agar. Hopefully you can kind of see why they call it branching. The little filament forms that it takes on. Kind of cute. All right, Nocardia will inhabit the soil and water. You'll get it through either inhaling the bacteria, it doesn't produce spores, or getting it through like a skin abrasion or something like that. It does have resistance to phagocytosis. It does produce different enzymes. It does produce catalase enzyme, that's why it's catalase positive, but also produces different hemolysins. In the case of Nocardia, it causes either pulmonary infections or skin infections. And then if it causes a disseminated kind of all over the body, that's where it gets to be very poor prognosis for that patient. For Streptomyces, that was the one that was non-acid-fast. Again, it is also found in the soil, same kind of transmission, and the disease, they call it Actinomycetoma, so it's big lesions on the skin for this. And there's kind of an example of what those skin lesions would look like. So again, Nocardia also causes skin infections and pulmonary, and then Streptomyces is known to cause those skin lesions called Actinomycetoma. So to identify, one, you're seeing these filament forms or hyphae extending into the air, those branching on the agar. That kind of is an indicator. You can gram stain it. If it doesn't gram stain well at all, maybe it's a Nocardia and you could acid-fast stain it, that kind of thing. There is a broth that can be used to help differentiate between them as well. So not a ton that I test you on this last group. This last group is a little tricky. It's just not very commonly found in clinical isolates. So it's not something I want you to know a ton of. Just keep it very basic with these last two that I mentioned. So, and then here's another flowchart example of all over how to work up gram-pos rods in general. So you can see how it splits it from non-spore forming to spore forming. Again, the only one we discussed that was spore forming gram-pos rod so far is Bacillus. And then it talked about hemolysis and motility. Remember Bacillus cereus, and there's some others that are beta hemolytic modal, whereas Bacillus anthracis was non-hemolytic, non-modal. And then it went over to the other side. We talked about all of these, except for maybe the Arcano one. Talked about if it branches versus not, catalase testing it next, and then some other things that we've discussed. So you can see how they've used some of those test results to help start to separate which gram-pos rod it might be. Okay. So really, that was a huge PowerPoint. I know you might feel overwhelmed, but when you think of it, there's only like how many bacteria, like maybe 6 or 7 total to know actually out of that whole thing. There isn't a ton of bacteria really when you think of it. It was 62 slides, but really there's like 7 bacteria to take away. So just take the information you need out if that overwhelms you to have this large of a PowerPoint. All right, that is it for this week. You guys have a wonderful week, and we will talk next week. Thanks.